It is absolutely amazing how much DNA you can extract from 3 little strawberries. And how important, this just shows how important DNA is to the functioning of every single living thing on the planet since the first life began. In the very beginning, organisms didn't have DNA, but they had small sequences of genetic information that allowed them to do a few simple things like feed, survive adverse conditions, and reproduce. Think about this. You're walking down Cary Street in Carytown, and you see a very attractive person walk by. Well, as a result of that perception, your body actually reacts in a physical, emotional way. And the crazy thing is this too, is that each one of us is attracted to different people, different types of people. So there's something going on here with our DNA that, based on our perception, changes how we act and changes how we feel. And this is all based on simple genetic sequences. And we're going to move backwards a little bit to the beginning of our biology class, just to give a little review of the important things that we need to know in order to really understand DNA. We're going to have a lot of fun in this class. We're also going to learn about some crazy ways in which DNA and genes and genetic material is manifested in your everyday lives. But in order for you to really understand DNA, we got to go back and understand what DNA is made of. In this section, we're not going to go and actually learn about what DNA is made of. We're going to save that for another time. Today we're going to learn about all the building blocks of DNA. So first of all, the smallest particle that we work with in biology is the macromolecule. All right? Macromolecules make up cells. All right? And cells are made of organelles. All right? So macromolecules are molecules that are made of many chemicals bonded together in a specific way, and these macromolecules make organelles which then make up cells. Cells, conglomerates of cells, make tissues, and tissues then make organs. Now organs, working with other organs, make up something called organ systems, and these organ systems are what make up the human being. Now where does all this start? It's all about carbon. Carbon is the basis for all life, and it builds and makes and is the foundation for every single structure that makes up a living thing. Now, all of these things, the macromolecules that make up the organelles, and the organelles that make up cells, and the cells that make up organs, and organs that make up organ systems, are all made of carbon. Now, carbon is really great because carbon is stable. All right? It has 4 valence electrons, which means it can fit 4 more valence electrons in its outermost shell. Because of these 4 electrons, it can actually make bonds with 4 different atoms. All right? So if you see here, all the carbon atoms in all of these pictures here, in all of these structures, carbon is always bonded, has 4 bonds. Whether it is bonded to 3 things with a double bond, all right, or whether it be this carbon is bonded to 2 other things with a triple bond, they're always going to be making, each carbon will always make 4 bonds. Now another great thing about carbon is, unlike oxygen and hydrogen, which are actually very electronegative, all right, or electropositive in the case of hydrogen, carbon is very stable. It doesn't pull electrons too closely or with too much power. And it actually isn't weak either. So carbon doesn't just let atoms make bonds with other things and break and make bonds, kind of like the hydrogen bonds of the water molecule. So carbon, not only is it stable, all right, it's also very abundant in nature. And because it's abundant, all living things have access to carbon. All right. As you'll see here, carbon can be arranged in a diversity of ways. All right. Here we have a branched carbon structure. We have a straight chain with a branch here and a branch here. Here we have a straight chain carbon. All right. We even have carbon chains with double bonds. All right. And we also have a ring structure here. So straight chain, branch, double bond, and ring are some of the major, most common forms that carbon takes. All right. Now, depending on what organisms need the carbon-based structures for, they will use these different arrangement and diversities of shapes. Oftentimes, ring structures, all right, show stability. And the function of ring structures is often stability. An example would be steroids that go through the bloodstream need to be stable molecules, so they are often ring structures. Branch structures, on the other hand, tend to often be energy sources where you can take off pieces of the branches of the tree without taking the straight chain. So you always have the foundation, the straight chain part, and the cell, organisms, will take off the branches when needed for energy. All right? So one of the most common themes in human genetics, as in all biology or any branch thereof, is that form is going to meet function. So here's 3 more examples of form meets function. This molecule down here, this is an organic molecule called glucose. Glucose has carbons, hydrogens, and these oxygens all over it. Glucose is a commonly used energy source that moves through the bloodstream. Well, often things that move through the bloodstream and are designed to move through the bloodstream are ring structures, because the bloodstream is an environment that often breaks things down into pieces. So when you see the form of a ring structure, these things in organisms, and especially humans, often move through the bloodstream. If we move up here, we have a much different form. Even though they're still just carbons, hydrogens, and oxygens, like down here, the different form meets their different functions. The function of this molecule, it's called a fatty acid, is to store chemical energy. Over here, these things, positive limonene and negative limonene, are actually something called isomers. If you'll notice, they have very, very similar structure, and actually they have the exact chemical formula, but different structure. If you see, these 2 groups up here are actually shifted and different than the groups over here. This isomer, all right, and an isomer is basically a molecule or compound that has the same chemical formula but different structure. This isomer right here smells and tastes like lemons. This isomer over here smells and tastes like turpentine. All right? So when you change the simple structure of a compound, you can change its entire chemical properties. So what else is different besides structure between organic molecules? Well, one of them is called functional groups. All right, there are some common functional groups. The hydroxyl, OH, an oxygen attached to a hydrogen. A carbonyl, a carbon attached to an oxygen with a double bond. A carboxyl, a carbon attached to an oxygen with a hydroxyl. An amino, a nitrogen attached to 2 hydrogens, and a phosphate group. Phosphate groups are negatively charged and highly reactive. Amino groups are also reactive because the nitrogen itself is very electronegative. It pulls electrons to it with a strong pull. A carboxyl group, on the other hand, tends to release this hydrogen into solution, so it tends to be something called an acid, which is why you often hear carboxylic acid. Carbonyls are very stable as they are. The carbon-oxygen double bond is very strong. And we have the hydroxyls. The hydroxyls tend to also be reactive because they are polar. The oxygen holds on to the hydrogen, albeit very weakly. And the oxygen also likes to interact with other things. 2 more functional groups that we have are methyl and a sulfhydryl. A methyl group is just a carbon with 3 hydrogens, and a sulfhydryl group is a sulfur group attached to a hydrogen. Now, here's the deal. Why do you need to know functional groups? Well, the only thing that really differs between one organic molecule to another is the different functional groups that it has. The first macromolecule is the carbohydrate. The carbohydrate always has a carbon-hydrogen ratio to 1 to 2 to 1, and its most common functional group is the hydroxyl group. So, if you see a molecule with tons of hydroxyl groups on it, it's going to be a carbohydrate, or in other words, a sugar. Sugars, all right, can be either ring structure or straight chain, but we will mostly focus on the ring structure. A monosaccharide is a sugar with 1 ring structure. A disaccharide is when 1 ring structure attaches to another, di meaning 2. And the most common disaccharide on earth is sucrose, a sugar, which is table sugar. And we have something called polysaccharides. We have many carbon-based ring-structured chains attached to one another. Now, again, these are carbohydrates, and their most common functional group is a hydroxyl. You're going to see carbohydrates in human genetics because the sugars make up the backbone of DNA and are very important in how DNA reacts with things around it. The hydroxyl group is also something that's going to interact with water. So carbohydrates, which we're looking at here, sugars, are mostly going to dissolve or interact with water, and they're called hydrophilic. Hydroxyl groups, again, if we go back here, all right, are the most common functional group in carbohydrates. They are hydrophilic, all right, and which means they interact with water. So the next major group of macromolecules are large molecules. The second one are lipids. Now, when we talked about carbohydrates, for the first one, carbohydrates are mostly hydrophilic, and their functional group is hydroxyl groups. Lipids, on the other hand, are hydrophobic. They do not interact with water, and they do not have hydroxyl groups. There are two major types, fats and steroids. Steroids are easy to see. They're made of 4 fused hydrocarbon rings, and they're very, very stable. Examples are steroids like testosterone, androgen, estrogen, and cholesterol. They often act as chemical messengers in the body which send signals and are made of 4 fused rings. On the other hand, fats are usually energy storage, and they make up cell membranes, and they're made of long chains. So you can see here that the hydrocarbons that make up fats have completely different structure than the hydrocarbons that are made of steroids. But they're both lipids because they both generally just have carbon and hydrogen. They have the occasional oxygen. You can see here that form definitely meets function because the function of this molecule is for energy storage and the function of this molecule is to be a chemical messenger. As a result, their forms are different. This is an example of some poison ivy here, and often you can take steroids to resolve a systematic or systemic reaction that may result from a really, really bad case of poison ivy. Often steroids, chemical messengers, are what are used to treat really bad cases of poison ivy. All right, so those are the lipids, all right, the second group. The third group are proteins. All right, proteins are these large structures that make up things like enzymes and transport channels, and they have many, many different functions in the cell, but mostly enzymes that are involved in supporting and acting as catalysts for chemical reactions. Now, proteins are these really long chains of amino acids. The building block of a protein is an amino acid. All amino acids have a central carbon, a carboxyl group on one side, which under normal conditions this would be carbon to oxygen, carbon to an oxygen, and imagine there's a hydrogen here, and an amino group on the other side, a nitrogen attached to 2 hydrogens, and remember we said the nitrogen likes to attract other things, so it's attracting this hydrogen, but imagine that it's actually not there. So the amino acid structure has a carboxyl group on one side, an amino group on the other, a central carbon, and then a R group here that's also called a variant group. Okay? This is the basic structure of an amino acid. If you put amino acids together, all right, you're going to end up with these long chains called polypeptides. Amino acids built upon one another make polypeptides, and polypeptides build on one another, and they make proteins, which are the functional form of amino acids and polypeptides. Proteins then can catalyze chemical reactions and all that different stuff. So the primary structure, all right, of a protein is the straight chain here. Okay. The secondary structure is when that straight chain begins to bend. All right. When it starts to bend in this way, this is called the alpha helix, and when it starts to bend in this way, it's called the beta pleated sheet. This is a more two-dimensional structure, whereas this helical structure is more of a 3D structure. So there are 2 ways that secondary structure can occur, and it's all based on the R groups, or the different groups in the polypeptide chain. Tertiary structure is when these secondary structures start to fold upon themselves. All right, and when you mix multiple tertiary structures together, like this one, there's 4 different tertiary structures, you end up with things called quaternary structure and functional proteins. All right, so the most important thing about proteins is that their building blocks are amino acids, and the amino acids bond to one another to form long things called polypeptides. And then these polypeptides can then form more complex structures. So we've got carbohydrates, lipids, and proteins. When we move over to nucleic acids, they're the fourth major group of macromolecules. We're going to spend a lot of time on nucleic acids because nucleic acids make up DNA and RNA, which then are going to really be the focus of this entire class. So we're going to spend a lot of time on nucleic acids. But nucleic acids are basically, there's two forms, DNA and RNA. They have building blocks, which are called nucleotides. A nucleotide is a sugar, a nitrogenous base, and a phosphate group. All right, which when you can attach many of these together to form long chain structures, which we call DNA or RNA. So these little nucleotides are the building blocks. Just as amino acids are the building blocks of proteins, nucleotides are the building blocks of nucleic acids or